Alright, so as a review of this week's lab, I'll go over what we did, as well as a couple of definitions that I covered in the lecture, as well as in lab. So, as a recap, we had 4 treatments that we were testing on our organisms, potato. So the first treatment, or variable that we were testing the salinity of water. So we had both freshwater jars and saltwater jars. We were also testing the variable of size. So we had both large and small wedges of potatoes that we were testing or experimenting on. So our hypotheses were that the organism was going to have a difference or an observed change in their mass throughout the course of the experiment. And then an alternate hypothesis that we were also testing was whether we could observe some noticeable difference or significant difference between the mass at the start of the experiment and at the end of the experiment under each of these variables. So once again, those variables were freshwater with a large potato piece, freshwater with a small potato piece, saltwater with a large potato piece, and saltwater with a small potato piece. So that's how we set up the experimental design, and those were the questions we were testing. The whole reason why we were testing for salinity is whether we could see whether this organism, this potato, was a osmoconformer or an osmoregulator. So if it was an osmoconformer, it would be losing weight in terms of mass as soon as it was dunked into the salt water, and then it would be gaining mass when it was dunked into the fresh water. So there's that concentration of solutes within salt water, and then there's that concentration of solutes within the body of this organism, within each cell. So if it's an osmoconformer, the water is going to flow against that concentration gradient of solutes. So if we have a higher concentration of solutes outside than there is inside this potato, inside these cells, then water is going to be flowing out of it. And so that's the case that we saw with our saltwater potatoes. So this organism is osmoconforming. One of the things that we missed when we have goldfish, which is why I was pretty disappointed that I couldn't find any, is that we missed actually seeing an osmoregulator control the amount of fresh water that's entering their body or leaving their body. So if we had goldfish and we measure the difference in mass between the start of the experiment and the end of the experiment, there's a noticeable, there's no noticeable change between those two measurements. So that goldfish can live in pure water. It can also live in pure or brackish water, and it will maintain that constant internal environment. So maintaining that constant internal environment is what we call homeostasis. And so this is important for almost all life. If I mentioned the example of pH in your bloodstream, if that changes just the slightest, it's extremely dangerous for us. Our bodies are 72% water, so bringing it back to fresh water. If we don't drink for 3 to 4 days, we can die. In fact, if you lose just 2% of your body weight in water, you actually start to feel nauseated, tired, fatigued, and that's one of the first signs that you're extremely dehydrated. And then from there it goes much more rapidly downhill until the point where you also croak and die. So maintaining a constant internal environment is extremely important for life. So that's why there's those 2 different characters that we talked about, the osmoregulators and the osmoconformers. Osmoregulators are found in fairly unstable environments. So I like to use the example of the short crab. It lives in a tidal area, so it has periods where it's going through high salinity. So in the middle of the day when the tide's gone out, it's trapped in these pools, and salinity is increasing to dangerous levels. And it's also dealing with periods of low salinity, so when the tides come back in and it's being supplied with this much lower salinity water. So it has to deal with those 2 tolerances, that wide range of tolerance. So it's constantly controlling the amount of fluid that's flowing into its body, or fresh water that's flowing into its body, as well as the amount of fluids flowing out of its body. The other example I gave was of a spider crab. So a spider crab lives in a very stable environment. Not much changes. The water temperature is near constant. The pH is near constant. Salinity is near constant. All those variables, environmental parameters, are unchanging. So this organism doesn't need to expend the energy to control the amount of solutes inside its body, the amount of salinity or the amount of fresh water that's entering its body, because it doesn't have to deal with those changes that a osmoregulator has to deal with. So, what else? Talked about the difference between diffusion and osmosis. So osmosis is what we're observing today, with the movement of water across a concentration gradient of solutes. So the solute in our experiment is the particles that are dissolved into water. So in this case, it was iodized salt, right there. And the solutes are dissolved in the solvent, and so we were testing our solvent, deionized water. So deionized water, or just water in general, is a really important molecule because it is what we would consider a universal solvent. So it dissolves most organic and inorganic substances, or solutes, particles that we need. So it's an important molecule, has really neat characteristics, but this is zoology, not biochemistry or general biology, so can't really go into that much further. Let's see, what else did I go over? That was roughly about it. Talked a little bit about excretory systems in some of the classes. So we talked about protonephridia, that's found in the phylum Platyhelminthes, especially in those planaria, so those little cuties, the ones with the 2 eye spots that we saw last week. So those protonephridia have a single opening, and so it's sucking this body fluid through an opening, and it's absorbing all of its nutrients that it's about to lose, while also getting rid of any excess waste, such as ammonia, and pumping that into the external environment using its flame cells. And so the flame cells are just these ciliated cells that are at the entrance of that opening to the protonephridia. So I gave the reason why, a hypothesis on why these protonephridia first evolved. The idea goes that these planarians were living in a hypotonic solution, so they had more or a higher concentration of solutes inside their body than there was outside their body. So in order to keep pumping that fresh water out of their body, so they didn't just bloat and burst, they started using these ciliated cells, the flame cells, and started pumping water outside their body. When they moved into isotonic solutions, where their solutes, the concentration of solutes inside their body began to match or become lower than outside their body, they had to reverse the process, keep solutes or water inside their body instead of having it drawn out. So from this system, we get the very first excretory system, and from there we get into more and more complex excretory systems. So next week you'll see the metanephridia of an earthworm, and the nephridia of, or you should try and see, the excretory system of a mollusk and the clams. And then from there we'll move on to the arthropods. So we have a crayfish dissection, and you'll see the antennal gland that it has. Later on in the year, when we get to the pig dissection, you'll see kidneys, and so those are just really derived structures that we have. All mammals have kidneys, and we use it to filter toxins out of our blood, hold on to nitrogenous waste until we can let it go at an appropriate time. So I mentioned nitrogenous waste. There's 3 different types. There's ammonia, urea, and uric acid. Ammonia is the most expensive, energetically expensive to, uric acid is the most energetically expensive nitrogenous waste to produce because it's taking ammonia and turning it into a less toxic substance. The second most energetically expensive nitrogenous waste to produce is urea. So urea has a little bit of water loss. It's used mostly in terrestrial organisms, especially terrestrial mammals. So there is some water loss, but it's not extremely energetically expensive to produce. Plus, it's relatively non-toxic, so we can store it inside of our bodies. The third most, or least energetically expensive nitrogenous waste is ammonia. So really that's just a pure nitrogenous waste from metabolic activities and such. And so ammonia is fairly toxic. It's extremely toxic in aquatic environments. So in freshwater, when you have a flowing system, aquatic organisms will just release ammonia from their gills right into the environment. They don't need to expend a lot of energy to transform it into a much more or less toxic form. So they just have to worry about water loss if they're in saltwater environments, but there are adaptations to cover that. So sharks have kidneys, so they're producing urea and storing it for a while before they release it into the environment. So they live in this water, they live in marine environments. So they constantly have to keep fresh water inside their body and then salt water outside, or have that fresh water, or to try and prevent that fresh water from being drawn outside that body by that concentration gradient. So they're storing that urea inside their body, and they're trying to extract as much fresh water as they can from their waste before they excrete it from their bodies. So uric acid is really only found in terrestrial insects, desert mammals, and reptiles and birds. So they're all trying to waste the least amount of water, especially with organisms found in the desert. So when it rains twice a year, or you have a very limited access to water, you have to extract the most amount of water you can from your food, and then not waste any. So you extract the most amount of water from your waste before it leaves your body. So they have these really complex kidneys to handle this process. So the other thing we were testing in lab, so we had both freshwater and saltwater, and we were also testing size of these potato chunks. So we had both large and small wedges of these potatoes. With that, we were testing the surface area to volume ratio of these potatoes and how it affects the rate of osmosis. For the most part, you guys had good data. There was a couple, there were a couple sections that had data that didn't fit the general trend. So the general trend is that organisms, as they're much smaller, or as they get smaller, they have a higher surface area to volume ratio, and so they're they have a higher influence of osmosis or diffusion. So as surface area increases, the surrounding water has much more area to work with in terms of drawing out fresh water or pumping fresh water into it relative to its body volume. So to give you just a brief summary, if you have a length of a cube, say the length is 2 whatever units, the area of that with that same length is going to be 2 squared, and then the volume is going to be 2 cubed. So with a length of 2, you have an area of 4, and then a volume of 8. So as you can see, volume increases exponentially, whether you have a length of 2, 4, 6, 8, 10. And so area's length squared, so that's going to increase exponentially. Volume is length cubed, so that increases much more rapidly than surface area increases. So I'd like to mention how this is why insects and planaria and everything that's small stay small, and why larger organisms that have able to overcome this surface area to volume relationship have gotten as large as they have. So planaria don't have a respiratory system, they don't have a circulatory system, so they're limited on the size. So they can only grow to as large as, so they can only grow to the point at which their cells no longer are diffusing oxygen deep enough into their body. So since they don't have that respiratory system, they're not pumping oxygen into their body, and they're not pumping CO2 out. They have to rely solely on diffusion, which is a very slow process. So that's limiting their size. They can't get oxygen deep into their body to perform any cellular functions. Once you get up to a much more much macro size, such as with humans or elephants or blue whales, we have a very complex respiratory system. We have a very complex circulatory system. So we're pumping oxygen into our lungs. So whenever we draw down our diaphragm, our lungs expand. A huge volume of air and oxygen, nitrogen, CO2 flows into our lungs, and then from there it's dissolved into our blood, particularly the oxygen, and then it's pumped throughout our veins and our arteries and goes to where it's needed at the rest of our cells. So we have this relatively complex way of overcoming that surface area to volume relationship. And that's what we were testing with these potatoes. So we were trying to see whether larger potatoes or smaller potatoes have a proportionally larger impact on the change of weight noticed. So for the most part, you all had really good data, a couple exceptions. So what you should have seen that smaller potato wedges, because it has a higher surface area to volume relationship, those smaller wedges increase much more, or increase or decrease, or what's the way to phrase that? Have a larger or smaller, have a much larger percentage change of their body mass, whereas the larger wedges of potatoes would have had a much smaller change in their body mass proportionally. So those were the two variables we were testing, both salinity and size. So once again, we had the treatments, freshwater and saltwater versus large and small. Usually in the past, I've had students go through a t-test, and so with a t-test, you're making, you also have to calculate standard deviations, and so you take each individual measurement that you observed, and then you subtract that from the mean or the average of all your data, and you do that for every single observation. Take the square root, and I believe you divide that by the mean, so, if I remember it correctly. And then you just calculate a t-test. So you take the means of either of the two groups, so freshwater or saltwater, or large or small. You take those two means, you take the square root, and then you divide by standard deviation of those groups, something like that. Either way, it takes a long time to calculate by hand, and we didn't have enough time to cover it in this lab period, so with only an hour. So we'll get to it sometime, probably in the behavioral lab or the hemoglobin lab. But for now, you don't have to know it. Next week is our first dissections. So we have both a worm and a clam. So we're representing two different phyla. So the phylum Annelida, or the segmented worms, and then the phylum Mollusca, or mollusks, clams, squids, that stuff. Remember to take the quiz. It's due Wednesday at noon. For my sections, Michael's sections, yours is due whenever Michael's assigns them. So take the quiz, watch the next video, the lecture video, read the lab manual. Yeah, wear closed-toed shoes, wear a mask, simple stuff. You should know the drill by now.